And we're going to start today, which is webinar number 6. Okay, so you can see my screen, right? So webinar number 6 is HPLC peak resolution. We're going to talk about why it is important and also when it is not important. And then go into something called the resolution equation. We're going to define what it is, which you can see on the right-hand side, the resolution equation. Okay, so here's my agenda for this morning. We start with the question, why is peak resolution important? Why are separation scientists in the world so obsessed with something called resolution? Why do we really need baseline resolution? And also the other side is when it is not important, when is it less important to have peak resolution? So there are circumstances that you might not need to have peak resolution, right? So we talk about both sides. Why is it important? And when is it not important? And then we are going to discuss the resolution equation, which is the equation here. Okay, you see you have 3 factors, right? A factor of efficiency, selectivity, and retention. Okay, now please note that the resolution equations, it really only works in isocratic separation. So it does not work in gradient separation, where it's much more complex, right? However, it is a good concept to learn chromatography. Okay, so we constantly talk about efficiency, selectivity, and retention. Okay, so we're going to use that resolution equation and really define what it means to the user, right? What does it mean to you and me when we're developing a method, right? Why do we need to learn a little bit more about that? Finally, does the equation really work, right? It is not an exact equation, it's an estimate. We're going to show you that it does work pretty well by showing you a case study where we do the calculation from this equation and then compare it to the measure value. We can measure resolution by this equation, right? We take the difference in retention time and we divide it by the average peak width at the base, right? So this is the equation for actually measuring a resolution. You can also get the resolution from your CDS, from your chromatography data system, right? We can calculate in many different ways. For most pharmaceutical scientists, we would use the USP resolution. Okay, we would talk about that later on. What is the USP resolution? But basically, this is the measure value from the difference of retention time divided by the average peak width. Okay, why is peak resolution so important? Why do we keep talking about resolution? Okay, so the first thing, HPLC is a physical separation technique for analyzing a mixture. So primarily, we use HPLC to measure the levels of different components in a sample mixtures. And I keep saying that everything in the world is really a mixture, right? You have a pure drug, but if you look at it carefully, it is a mixture. It might have trace impurities and degradation products. Now, separation scientists are obsessed with resolution. Actually, we are paid to achieve resolution for a sample, okay? Oh, why? Because any co-elution between 2 peaks would lead to inaccurate data. Now, here's kind of assumption, right? Most pharmaceutical analysis use UV detection. So this is the caveat. This is the reason why we are obsessed with peak resolution. Okay, a UV detector used in most QC application. And this is just the facts of life. Okay, I don't think UV detector is such a great detector, but it is very precise. And it is the standard detector in quality control. It's not going to change in the next 10 years. It's not going to become mass spectrometry, right? Because mass spec does not have the precision. However, the UV detector would respond to any component that have any absorbance at that specific monitoring wavelength. So this is the caveat. This is the reason why we have to physically separate all the component in order to get accurate quantification, right? But it also depends on the intended application. See, that's kind of, everything has exceptions. So you really have to understand in a little more depth what we are talking about. So the gold standard or the expected resolution is 1.5, which stands for baseline resolution, baseline separation. You see a little chromatogram here, right? When you achieve a baseline resolution, you get most of the area underneath the curve. And there's a little bit of overlap, which is less than 2%, right? So that's actually pretty good. Now, it is a requirement in something called the regulated testing. Regulating testing, meaning that the testing is really important and is also regulated, meaning that being controlled by a regulatory agency, such as the US FDA, Food and Drug Administration, or EMA, European Medicinal Agency, right? So this is the two regulatory bodies in Europe and in America. For people doing generic drugs, probably the USP, United States Pharmacopoeia, is kind of like a regulatory agency, but it's really not a true legal entity, right? It is a consortium that everybody in generic really have to conform to. Is a monograph. If you have to say that your OTC drug, over-the-counter drug, is USP. If you want to label USP, you have to use their monograph. Okay, now here's the question: when is coelution or partial coelution acceptable? I'm talking about resolution less than 1, which is not baseline. When is it not important or less important? So I can see two circumstances, right? So first, in UV detection, a coelution is acceptable if the other interfering component has no absorbance, right? So even though it's physically together, it's okay because the other component is not absorbing, right? So essentially, your detector is selective at that monitoring wavelength to that particular component. So I would show you one example. I would not explain the method development of that example. It's actually a very difficult, challenging method development, right? I'm going to show you something which is pretty nice, but we will not explain it later on. We will talk more when we get into methods development, right? So how do we develop complex method? The second scenario is mass spec detection. Okay, so mass spec is actually a very specific detector, right? You can actually have it respond to only the parent ion, right? So for most basic components, the M+1 parent ions. So if you can use a mode called single ion monitoring, or SIM, or SIR, okay? So when you are in that mode, you can quantitate with a calibration standard using mass spec, which work really well, except for an isomer. If you have an isobaric compound, mass spec is not good. You still need an HPLC to do your separation, right? So that's kind of the caveat or the exception, right? So mass spec is definitely great, but mass spec is not really QC compliant. Mass spec is very difficult to have very precise quantification. You really struggle for mass spec to quantify down to 1%, which is not good enough in quality control of drugs for drug substances. We really need a precision less than 0.5%, right? So mass spec is really not acceptable for very precise QC work. So UV detector is still the king, the workhorse in quality control. Now I want to show you this complex chromatogram. Okay, it is a very difficult method development process, which is something I developed about 10 years ago, right? So at that time, I worked for a pharmaceutical company that actually manufactured this OTC product. OTC product means over-the-counter product. It is a laxative from a natural material. Now, for most of us that is used to Western medicine, which is using a pure API, active pharmaceutical ingredient, you're not used to a natural component. Natural components are problematic they're very difficult methods for separation scientists because they are very complex. It does not really have only one API. Any natural components, some of them might have hundreds of APIs. So to calculate the potency is not trivial, right? So for this Senosides from Senna plant, from a plant, it is a natural laxative. So this company that I work for, we market a product which have a sales of $100 million from a natural laxative, from a natural product, right? So in order to calculate potency, I have to sum up 8 Senosides. Okay, you can see the green ones. The major one, and Senoside B and A, right? So you have to actually calibrate each component, add them up to find your potency. It's a very difficult separation problem. Okay, and the UV spectrum is also very unique, right? The Senosides, this is Senoside A. You have 2 peaks. You really have 3 peaks, but this 212 is not a real peak. It's a quasi peak. Okay, it's not a real peak. You shouldn't use 212. So either 268 or 338 or 340, right? So there's 2 lambda max of the UV detector, right? So this is 2 monitoring wavelength you can use. So we have many products for this OTC laxative. We have a syrup product. We also have a tablet and a capsule. So after several weeks, I developed this method, right? And this method can actually calculate the potency by adding up 8 of the Senosides, right? So you can see that the monitoring wavelength is 270 using the first lambda max. And it works really well, right? You can see that even D1 and D are totally separated, and they seem pretty pure, right? It is really difficult to make sure everything is pure. When we come to the syrup, we have a big problem because the syrup, we need preservatives. The preservative in the syrup is 3 different preservatives: sorbic acid over here, and methylparaben here, and paraparaben here. In a syrup, ideally use the same method for the tablet and the syrup. But in the syrup, there is a problem because we cannot have the preservative will interfere with the potency assay, right? So here is the dilemma, right? Do I redevelop the method, which is very difficult, right? Or can I have another way? So the way I actually choose to have the syrup method is actually change the wavelength, right? To use the wavelength at 340. Where the preservative does not have any absorbance, right? So all I have to do is to redo just the relative response factor, and then I actually can have a reasonable potency for the syrup, right? So sometimes being a separation scientist, you want to be flexible. You want to do something very quickly, but accurately, without harming the timeline for the project, right? So this is kind of a scenario. Later on, we would get into the method development and show you. Now, this actually is a very nice separation. It's using a HPLC column. This particular column coming from Waters Corporation, I have 80,000 plates, right? It is definitely a single column. It is 30 centimeters long, 2.1 millimeter ID, packed with sub-2 micron particle. This is a very nice column. It's very long, give you very separation. However, the back pressure is very high, so I can only use 0.3 mil per minute, right? So this is definitely using HPLC to the max to give you very high resolution. So hopefully you like this chromatogram, and you appreciate that sometimes we don't have perfect method, right? So we have to compromise, and there are ways to make your life simpler by understanding what the goals of the methods are, right? So by changing the lambda max, the monitoring wavelength from 270 to 340, we can have accurate quantification without interferences from the preservatives. I hope you like this case study, right? It's kind of, it's one of my better method development application. Our second case study is using LC mass spec, right? This is a generic method that I have been advocating. It is a generic method that seems to work for every API, every NCE in this particular company that I work for. Okay, so that was published. It's called a universal generic reverse phase method for pharmaceutical analysis. It is a 2-minute method. Oh, I'm sorry. Okay, this one's a little longer. This one I try to get a little better separation. So the generic method is 2 minutes, and you can extend it to about 8 minutes, 7 or 8 minutes using this condition. Okay, and when you do this condition, you actually can separate all 12 NCE, new chemical entity. You can see that I have a mass spec detector from Waters. It's called the QDA. It is a very simple, compact mass spec. And the software actually did a very good job. It can have thumbnails of all the UV spectrum, and all the mass spectrum are being drawn into one report, right? You can see that, all the mass spec are here and the UV spectrum are here, right? And this is a separation at 255, right? Now, one of these nice things that you can have using a Waters and power detector is to automatically extract a single ion chromatogram, right? So this is called an extracted ion chromatogram, right? So you can see that for every of the parent ion, there is an extracted mass chromatogram. So let's say, looking at this pair of peaks here, right? So you have a little bit of co-elution. It's almost baseline, but not quite. However, the automated extracted ion chromatogram, it is unique to that ion, right? So you can have very specific detection, even though that if these two peaks are co-eluding it, using mass spec, I can get a reasonable estimates. Well, actually quantitation using mass spec. However, we don't use mass spec for QC, right? Okay, so we talk some more when we come to mass spectrometry. It's very important for separation scientists to really understand mass spec, because that's the wave of the future. Okay, so let's talk about the isocratic separation, the resolution equation, right? And what are some of the implications, right? So here's the resolution equation. The resolution equation is a quasi-quantitative equation. The resolution equation defines the resolution that can be achieved from 3 different major factors. Factor efficiency. Please note that the efficiency, which is defined by the plate count, and the big N is the plate count. Now, we have not really talked about plate count so far, right? So that is in the next few weeks, we talk about plate count. Right? You have a better understanding of plate count then. However, things are never perfect, right? So we have to kind of describe it now. So the plate count, the relationship is the square root of the plate count, right? So it's not a straight proportionality. For retention under isocratic condition, we have K. K is the retention factor, and the factor actually is K over K plus 1, right? So kind of very strange factor here. And also we have something called selectivity. Remember, selectivity is the ratio of the 2 closely eluding peaks, right? Alphas are always close to 1, right? Because we only care about peaks which are closely eluding, right? So they're always near 1, right? So alpha minus 1 over alpha. Alpha is always near 1, right? It's the selectivity factor. Now here I want to define the goal of most LC separation. The goal of most LC separation is to achieve resolution for all key analyte. Now here is the caveat. Here is an important thing. It's called the key analyte. What is a key analyte? If you have an assay for just potency for the main component, the only key analyte is the potency. You don't care about the degradation product. You don't care about anything else. If you have a dissolution assay, all you care is about the release of the API. So you don't need an assay for the API only, right? So in this case, the intended analyze only the component. Now on the other side, you have purity assay, you have stability indicating assay. That is really important, right? We want to know everything about a sample to protect the patient's safety, right? We want to know that the drug substance or drug product contain only the API plus a limited amount of impurity and degradation product, right? And we know the shelf life of your drug product after doing a lot of stability study. So the key analyze is really everything, right? So you want to make sure everything is eluted. You want to detect the API, separate it completely from all the process impurity and degradation products. So ideally, we have a resolution of baseline, which is 1.5. Now, on the other hand, if you are a bioanalytical chemist, a PKDM, pharmacokinetic, or drug metabolism, you're measuring drug in serum or in serum or in plasma, in human sample, or in a rat sample, or in cyno or monkey sample, right? So that's your job. Now, all we care are the drugs in the physiological fluid. We don't care about protein, albumin. We don't care about all the hundreds of thousands of proteins in the serum or plasma, right? So, I mean, there's a lot of things in there, but we don't care about those things. All we need to do is to measure the drug and its metabolite. And in those cases, you really have to use LC mass spec. Actually use LC mass spec, right, triple quad, to get you selectivity. So once again, the goal of the separation in those cases is to quantitate your drugs only, right? You don't care about the extraneous component. You don't care about the caffeine if the patient have a cup of coffee. If you use UV, you see a big caffeine peak, but you don't care about caffeine. We only care about the kind of drug that the patient has taken. Okay, maybe no, after dosing. Okay, so the three efficiency factor, right? So N is efficiency. Now, I want to define what N is. N is the column length, typically divided by twice of that of the particle size, right? So we talk about this equation later on, what constitutes this equation, right? This is an estimate. So if you have a very complex sample, you want to have a longer, high efficiency column packed with small particle, so that you have a large number of plate count to give you the maximum resolution for overall. K is also dependent under isocratic condition on the percentage of mobile phase B, or the percentage of acetonitrile or organic solvents such as methanol or acetonitrile in your mobile phase, right? And the factor is K over K plus 1, right? So this factor is approaching 0 when the retention factor of the last peak is close to 10, right? So it's already 0, right? So by more retention under isocratic condition, it doesn't buy you much, right? Because if you have a retention factor of 100, it's actually no good because the peaks are so broad. Because under isocratic condition, the peaks do get broadened with time. Finally, alpha or selectivity is really important, and it's dependent on the type of bond phases, chemistry, depending on the mobile phase A and B, right? So these are the examples. So when we were doing method development for stability indicating method, for critical method, we always focus on changing selectivity to achieve baseline resolution of all key analytes in the sample. So that almost means everything for a drug substance sample, right? So those are really difficult method to develop. Typically take you at least 2 weeks, sometime up to 3 or 4 weeks to develop this type of method, right? You never have 2 years to develop a method, right? Okay, so you have seen this application before, right? So this is the effect of changing K by lowering the solvent strength, right? So if you develop isocratic method, one of the best way to do it is to inject the sample of your mixture under 100% acetonitrile. So typically you start with 100% acetonitrile. And then in most cases, there's no retention. Everything we elute at the solvent front, right? Now, and you keep dropping the mobile phase strength down to 45% acetonitrile. And for this particular sample, which consists of 7 substituted phenols, so I have a total of 7 substituted phenols. So I actually weigh 7 phenols into this mixture, right? So I expect to see 7 peaks, right? At 45% of acetyl nitrile, I have 5 peaks, right? So I know I'm getting better, but not good enough, right? So I continue to drop the solvent strength to 35%. And here I can see that I have a pretty decent method, right? The only thing not ideal is a resolution between 1 and 2, right? So this is almost a good starting point, right? Now the K prime of the last peak is K prime of 5, right? You can see that K prime is still not too bad. By going lower in solvent strength to 30% acetyl nitrate, I can see that I achieve a very good separation of all 7 peaks, right? So from methoxyphenol, cyanol, phenol, fluorophenol, nitrophenol, right? Everything is separated baseline. So this is almost a perfect method, a good starting point, right? Then I can fine-tune the method to make sure I have sufficient sensitivity, right? Now, this is not a common application. In pharmaceutical company, we don't do this type of thing. This is really for column manufacturer, such as Sopelco, to do that, right? So we never, in our real life, do this type of thing. This is kind of fun, but we don't do this type of thing, right? With the exception that you're looking for water-soluble vitamins or something like that, right? But in real pharmaceutical development, we always have a single API peak plus a lot of process impurity and degradation product peaks, which have not been defined if you start from scratch, right? If you start from early development, you don't really know what it is. And we always, without exception, use a gradient in those type of very critical method, right? But here is an example of isocratic separation, a method development. We call this sequential isocratic step, right? So you want to have a potency method for dissolution method for a rough potency. If you want to measure the stability of a dosing solution for a NCE, a new chemical entity, you would use this isocratic method. Okay, you've probably seen this before, right? So this is the effect of solvent type on alpha, on the selectivity, and also eventually on resolution, right? So here we have 3 mobile phase B under reverse phase, which is acetyl nitrile, methanol, and THF, right? And here I have a pseudo sample. This is a 2-component sample. At 60% water with 40% acetyl nitrile, the alpha is 1.07, and I don't have, the resolution is about 1 or a little bit less than 1, right? So this is not ideal. This is not good. I can go to methanol. I have to use more methanol because methanol is weaker. And the alpha is much better, it's 1.2, right? You can see that a little bit change in alpha from 1.07 to 1.19. You can achieve quite a bit increase in resolution, right? Why is it so? Remember the resolution equation, right? Alpha is always near 1, right? So if I increase alpha from 1.1 to 1.2, I double the resolution, right? So a little bit change in alpha by changing the mobile phase B type of organic solvent, I give you a lot of changes, right? So particularly in this case, right, going to methanol is good. Now methanol is really good for isomers because methanol can do hydrogen bonding with the analyte, right? So acetyl nitrile does not do hydrogen bonding. Now THF is actually even better, but we don't use THF because it's too toxic. It is really bad for your body, right? So we try to get away and not using THF, tetrahydrofuran, right? So we talk about that in webinar number 4. Here's a resolution equation, okay? And here is how they change, how by changing alpha, N, and K would affect resolution, right? So the first thing is that K is really not too great, right? Now, obviously, the K cannot be equal to 0. K equal to 0 meaning that is all the analytes eluting with the solvent front, right? So you're not doing chromatography, you're doing flow injection. There's no retention of your analyte, meaning that there's no separation. Right? There's no retention, right? How can it separate? And So K under isocratic condition, the factor is K over K plus 1, right? So when K is equal to 10, it's already approaching unity, right? So going to K equal to 20 does not buy you much, only broader peaks and less sensitivity, right? So K is not an important factor if it's more than 2 or 5, right? Now alpha is really great, right? We say alpha is have a lot of impact, right? So so typically when we do method development, we do selectivity tuning. We keep changing things, factors, until we have all the critical pair of peaks resolved. So whether you know it or not, when you do method development, you're doing selectivity tuning. You try to change the alpha to your advantage. And also, resolution is proportional to the square root of n. So you can see the tendency of n, right? So by using a longer column, by doubling the length of the column, we can increase resolution by a factor of about 30%, right? Because the square root of 2 is 1.33, so about 33% increase in resolution, right? Now under isocratic condition, right? Under a gradient condition is kind of similar, right? The estimate, also the resolution is probably proportional to the square root of n. But you pay for it because then your analysis time is at least twice as long, right? So you might, your analysis time is longer. Okay, so how do you calculate resolution from the resolution equation, right? So here's one example, right? So in this example, which you've seen before on selectivity, so we have a pair of peaks, and alpha is the ratio of the K prime. The K prime of the first peak is 3, second one is 3.7, so the alpha is 1.30, right? So this is the alpha. Okay, so we can show you that the resolution equation does really works, right? Because we can actually do the calculation from the resolution equation from measuring from these parameters, right? So we know what the K is for each peaks. We know what the alpha is. The alpha is 1.3, and we know the size of the column, right? So the size of the number plate count of the column is 5,000, right? So we can do this calculation. We can calculate that the resolution from the equation is about 3.0, which is about right. It looks right? It's better than baseline resolution. We can also measure the resolution by the differences of the retention time divided by the average peak width, right? So if I do the calculation, do the measurement and the calculation using the resolution equation, using the calculation, I get a resolution of 2.8, which is very similar to that of the resolution equation, right? So just to demonstrate that this quasi-quantitative equation does work in real life, right? Using an actual measure example. Okay, finally, I want to show you one new information on system suitability, right? So every time you do regulatory analysis to do quality control, you have to demonstrate that your system or your method is suitable on that day, right? So anytime you do pharmaceutical analysis, you have to do about 10 samples of system suitability testing. Now in the USP and also in the US FDA called CDA, CDA stands for the Department of Drug Evaluation and Research, right? So this is a regulatory body from the US FDA. So you often hear these three common system suitability criteria, right? So you all often hear that FDA would like you to have a resolution of 2 for your key component, right? So this is ideal. Sometime it might not be possible, but that is something that you want to achieve if you can. Also, the peak tailing, the USP peak tailing factor should be less than 2, right? So we often use this type of criteria. And the repeatability actually is, most people use less than 2% RSD. for peak area repeatability. Now, this is actually not good. This is too wide, right? But everybody used that because that was cited in the USP and also in CEDA, right? For 5 replicates, you want to demonstrate the precision of your system, right? Your system precision, your auto sampler have to be less than 2%, which is too easy, right? It's really not a good criteria. So the reality when you are in new drug development for modern NCE, most modern NCE are very much complex. They are usually very basic. A lot of them have multiple chiral center. So in reality, the system suitability criteria can be very different, right? Now, here are some of my thinking. Now I'm not the US FDA, right? But I have done, quite a number of projects, right? So this is my experience. What is the reality of system suitability? So while this is great, a lot of time it's not possible to achieve that, right? So often, if I am lucky, I get a resolution of 1, right? Yeah. And if you cannot get 2 or 1.5, you really have to justify it in your method development report, right? To make sure that you've done enough, right? And sometimes when you have very complex drugs. Okay, so next week, I'm going to show you a very complex drug, a drug product with multiple API, right? So this one example I would show next week come from Gilead Science. A very, very difficult separation, take them years to develop. And they did not achieve a resolution of 2 because the marker solution for SST to demonstrate resolution contains 60 components, right? This API, this drug product have 4 API, is the standard of care for HIV, which then for ACE is an ACE drug, standard of care for ACE drug, right? So it's nice to see actual case study of the reality versus what is ideal. Okay, so the rest of the tailing factor ideally should be less than 2. But in most of my NCE that I have experienced, because they have multiple amine in the drug, they have a different scaffolding, having primary and secondary quaternary amine. It's actually some of the tailing factor are actually much worse. That can be as much as 4, right? Particularly if you overload the drug, right? The API peak should be more than 1.5 absorbance unit or 2 to give you sufficient sensitivity to go down to 0.05% sensitivity, right? So we need that type of sensitivity for stability indicating assay, right? So sometime you have to say that my system suitability factor have to be 4, right? Now, obviously you cannot be 10, right? Because that would not be good. The repeatability is 2% or 1%. You've seen a lot of them, but really in the latest monograph from the USP, it actually mentioned a better criteria, which is 0.73. This have a little bit more statistical sparing, right? So this is actually a better term, right? And actually, I would say that 0.5% is even better. It's more realistic because if you have a autosampler problem, right? If you have bubble in your sampling syringe, for instance, right? And you cannot get 0.5%, then you should know, you want to know that your system is injecting properly. So this is actually a much more useful SST criteria than 2%. 2% is really too wide. Why? Because your specification for your drug substance, the specs for drug substance is 98% to 1 or 2%, right? So the specs for the drug substance is actually 2%, right? So if your HPLC sampler is only capable to do 2%, it is not good enough, right? So definitely 0.5% is even more realistic, right? So for SST, I did publish something almost 20 years ago. This is an ACS journal for today's chemists at work. It's now defunct. It is not in circulation anymore, right? But anyway, that was a paper written quite a while ago. Talk about system suitability testing. Okay, so in summary, the resolution of key analyte is the most important for accurate quantification using UV detection. We have discussed a resolution equation under isoquadratic conditions based on three factor, right? Which is K, alpha, and N, right? So K is K over K plus 1, alpha minus 1 over alpha, and the square root of N. So changing selectivity, because selectivity tuning is usually the best way to resolve too closely eluding peak. That means changing the column, changing the mode for A or B. So we talk about method development later on. Okay, so in this webinar, we illustrate how resolution equation can be used to calculate resolution. And it's actually a good match from the measure resolution. Okay, so finally, we say that for very critical method using gradient analysis, the resolution equation really does not apply because it's very complex, right? So there's really no K in gradient. You also cannot measure plate count under gradient condition, right? So this equation is really not for gradient separation, where the critical method, uses gradient separation. So, however, the concept's really nice to learn, and since the gradient separation, N, K is no longer useful. K becomes very complex, and you almost have to understand differential equation to understand the average K prime for a single peak, right? So it is something that practically is not useful for most people, right? Unless you're really deep into the chromatographic theory, right? So in practice, they are not that useful. Okay. So here's the three references. Right? FDA probably is okay with it, right? Obviously, you have to try, right? You can't say that I can't get it. You have to document what you did, right?